Hi everyone, welcome back to the Science Box. So today I'm going to be telling you everything you need to know for the Biological Molecules chapter of AQA A Level Biology. The timeline's in the description, so if there's a particular part you're stuck on, feel free to just skip to there. So a polymer is a long chain of repeating subunits called monomers. This is kind of like a necklace where each individual bead is a monomer and the chain as a whole is a polymer. Between each pearl is a bit of connecting string, and this is the bond between the monomers. So for carbohydrates, the monomer is a monosaccharide, the bonds between the monomers are glycosidic bonds, and the polymer made up is a polysaccharide. For proteins, the monomer is an amino acid, the bonds between the monomers are peptide bonds, and the polymer made up is a polypeptide chain. And for nucleic acids, the monomer is a nucleotide, the bonds between the monomers are phosphodiester bonds, and the polymer made up is a polynucleotide. We'll get onto lipids later because they're a unique polymer in that it's not one repeating monomer, it's a combination of different monomers. For triglycerides, for example, which are a type of lipid, the monomers are both fatty acids and glycerol. To make monomers into polymers, we use condensation reactions, in which two molecules are joined together with the formation of a chemical bond and the production of a water molecule. To break down polymers into monomers, we use a hydrolysis reaction, in which a molecule is split into two smaller ones using a water molecule. Okay, so we're going to start with carbohydrates. A single monomer of a carbohydrate is called a monosaccharide. A pair of monosaccharides can be combined to form a disaccharide, and monosaccharides can be combined in larger numbers to form polysaccharides. A monosaccharide that we already know of is glucose. Glucose has two isomers, alpha glucose and beta glucose, and you have to be able to distinguish these structures from one another. The difference is, in the alpha isomer, the OH group points downwards, and in the beta isomer, the OH group points upwards. The way I remember this is with the phrase beta up, a bit violent, but there we go. So all monosaccharides and some disaccharides, like maltose and lactose, are reducing sugars, but sucrose isn't. To test for a reducing sugar, we use the Benedict's test. So first we add the dissolved food sample, and then we add the Benedict's reagent. Heat the test tube in a water bath, and if reducing sugars are present, then a brick red precipitate will form. So, as we said previously, disaccharides are monosaccharides combined in pairs. Glucose plus glucose equals maltose, glucose plus fructose equals sucrose, and glucose plus galactose equals lactose. Just a reminder that these reactions are all condensation reactions and the bonds between the monosaccharides are glycosidic bonds. Okay, now we're going to talk about polysaccharides, the first one being starch. Starch is a storage compound in plants and not animals. Especially large amounts occur in seeds and storage organs such as potato tubers. Starch is made of chains of alpha glucose subunits. These chains may be branched or unbranched. The unbranched chain is wound into a tight coil that makes the molecules very compact. We need to know how this structure of starch makes it suited for being a storage molecule. So firstly, it's insoluble, therefore doesn't affect the water potential of cells, so water isn't drawn into the cells by osmosis. It's also large and insoluble, so does not diffuse out of cells. It's compact, so a lot can be stored in small spaces. When hydrolyzed, it forms alpha glucose, which is both easily transported and readily used in respiration. And also, the branched form has many ends, each of which can be acted on by enzymes simultaneously, meaning that glucose monomers are released very rapidly. To test for starch, we use iodine, which is straw-coloured, and changes to blue-black when added to the sample if starch is present. Okay, now moving on to glycogen. Glycogen is a storage compound found in animals and not plants. It's got a similar structure to starch, but with shorter chains and is more highly branched. And just like starch, it's made of alpha glucose. So here are the reasons the structure of glycogen is suited for storage. It's insoluble, therefore doesn't affect the water potential of cells. It's large and insoluble, so does not diffuse out of cells. It's compact, so a lot can be stored in small spaces. And it's more highly branched than starch, so more ends can be acted on by enzymes simultaneously, meaning that glucose monomers are released very rapidly. Okay, so the final polysaccharide you need to know about is cellulose. Cellulose is found in plants and not animals. Sorry, that's wrong on the screen there. Cellulose is made from monomers of beta-glucose rather than alpha-glucose. It's made of long, straight, unbranched chains of these monomers which run parallel to each other, allowing hydrogen bonds to form cross-linkages between adjacent chains. The structure is suited to its function of providing support and rigidity because, firstly, the chains are cross-linked by hydrogen bonds, which adds collective strength, and then also these chains are grouped to form microfibrils, which are grouped to form fibres, which adds more strength. Okay, now we're going to move on to lipids. So, two groups of lipids are triglycerides and phospholipids. A triglyceride is made up of one glycerol molecule and 3 fatty acids, hence why it's called a triglyceride. And a condensation reaction happens between glycerol and each fatty acid, and an ester bond connects the two together. Fatty acids can be unsaturated, meaning that there are one or more carbon-to-carbon double bonds, or saturated, meaning that there are no carbon-to-carbon double bonds. Lipids are insoluble in water but soluble in organic solvents like alcohol. So here are the ways the structure of triglycerides relate to their properties. Firstly, they have a high ratio of energy-storing carbon-to-hydrogen bonds to carbon atoms, making them an excellent source of energy. They have a low mass-to-energy ratio, so are good storage molecules because energy can be stored in small volumes. They're insoluble, so don't affect water potential and movement of water by osmosis. And they also have a high ratio of hydrogen to oxygen atoms, so release water when oxidized, therefore are also a source of water. Okay, now moving on to phospholipids. Their structure is similar to lipids, but one of the fatty acid molecules is replaced with a phosphate molecule. The fatty acid molecules are hydrophobic, so repel water, and phosphate molecules are hydrophilic, so attract water. So phospholipids have a hydrophilic phosphate head that interacts with the water and a hydrophobic fatty acid tail. Molecules that behave this way are said to be polar because one end behaves differently to the other. And here's how the structure of phospholipids relate to their properties. Like we said, they're polar molecules, so form a phospholipid bilayer within the cell surface membrane. So a hydrophobic barrier is formed between the inside and the outside of the cell. Also, the phospholipid structure allows them to form glycolipids by combining with carbohydrates within the cell surface membrane. To test for lipids, we add our food sample to a test tube and then add ethanol. Shake the tube to dissolve any lipid in the sample. Add water and shake the tube gently again. A cloudy white emulsion indicates the presence of a lipid. Okay, now moving on to proteins. So, as we said before, the monomer for proteins are amino acids. A pair of these amino acids are called dipeptides, and a chain of amino acids makes a polypeptide chain. A functional protein may contain one or more polypeptides. Every amino acid has a central carbon atom attached to 4 different groups: an amine group, a carboxyl group, a hydrogen atom, and an R group, which is different for every amino acid. The structure of a protein has 4 levels. The primary structure is the sequence of amino acids. The secondary structure is the hydrogen bonds that form between the amine and carboxyl groups, causing the polypeptide chain to be twisted into a 3D shape of either an alpha helix or a beta pleated sheet. The tertiary structure is the 3D tertiary structure formed by interactions between R groups, and the quaternary structure is the interaction between polypeptide chains. The test for proteins is the burette test. Put the food sample into a test tube and add burette reagent. A positive result is the solution going from blue to purple. Now we're going to move on to a specific type of protein, enzymes. Enzymes lower the activation energy of the reaction they catalyse. Enzymes have an active site with a specific 3D tertiary structure which is complementary to the substrate. They then combine to form an enzyme-substrate complex. Those phrases in bold are mark scheme phrases that come up time and time again, so definitely memorise those ones. So at GCSE, we learn the lock and key theory, but this is quite a simplistic model. At A level, we used the induced fit model to better understand how the enzyme and substrate fit together. So this is the induced fit model of enzyme action. So the substrate binds to the active site, and the active site changes shape slightly, distorting the bonds in the substrate, so the substrate is complementary to the active site. The strain distorts the bonds in the substrate molecule and therefore lowers the activation energy needed to break the bond. This is the effect of pH on enzyme action. So pH affects how enzymes work because, firstly, the change in pH alters the charges on the amino acids that make up the active site. Therefore, the substrate no longer becomes attached to the active site. And significant changes in pH may cause the bonds maintaining the enzyme's tertiary structure to break, so the active site changes shape. Okay, so there's two types of enzyme inhibition, competitive inhibition and non-competitive inhibition. We'll start with competitive inhibition. So the shape of the competitive inhibitor is similar to the substrate, therefore the inhibitor can compete with the substrate to occupy the active site. The inhibitor is not permanently bound to the active site, so when it leaves, another molecule can take its place. If substrate concentration increases, the effect of the inhibitor is reduced, because the substrate molecule is increasingly likely to occupy the active site. At a high enough substrate concentration, the substrate molecules will occupy all active sites, and the competitive inhibitor will have no effect. In non-competitive inhibition, the inhibitor attaches itself to the enzyme at a binding site which is not the active site. Upon attaching to the enzyme, the inhibitor alters the shape of the enzyme and active site, so the substrate molecules can no longer occupy active sites, therefore the enzyme cannot function. Increases in substrate concentration do not decrease the effect of non-competitive inhibitors. And finally, I just wanted to add this because I've noticed it come up in exam questions a lot. So if you're asked to end any enzyme-controlled reaction, you can either boil the solution or add a strong acid or alkali to denature the enzyme. The only practical associated with this topic is required practical 1, which is the investigation into the effect of a named variable on the rate of an enzyme-controlled reaction. The independent variable for this experiment is temperature, the dependent variable is the rate of reaction, and there are multiple controlled variables, so for example pH, enzyme concentration, and substrate concentration. The enzyme we'll be using is trypsin, which is a protease, so digests proteins. One protein it digests is casein, which it breaks down into smaller, more soluble peptides. And casein is found in milk. We're going to start by drawing a cross on two test tubes. One of them will be our control, we're going to call that C1, and one of them will be our main experimental tube, and we're going to call that T1. We don't put any trypsin solution into the control to prove that trypsin is needed to carry out the reaction. Now to both tubes add 10 centimeters cubed of milk powder solution. Milk powder contains our substrate casein, so we need to keep the concentration of our milk powder solution the same. Next, grab two more tubes. To one of these, add 4 centimeters cubed of pH buffer. This tube is part of our control experiment, so we're going to call that tube C2. And to the other tube, add 2 centimeters cubed of pH buffer and 2 centimeters cubed of trypsin solution, and we're going to call that tube T2. Incubate all 4 tubes at 20 degrees Celsius for around 5 minutes in a water bath. Next, add the contents of C2 to C1 and the contents of T2 to T1 and start a timer. Time how long it takes for the cross to become visible. This happens because the trypsin breaks down the casein, causing the solution to go colourless. Our control doesn't contain trypsin, so the cross on the control tube should never become visible. Do the exact same experiment again, except incubate the test tubes to 30 degrees and then repeat for 40 degrees, 50 and 60. Now I'm going to give you some example results. We're going to do 1 divided by the time to find the rate of the reaction. Notice how the rate has the units s to the minus 1 because 1 divided by s is the same as s to the minus 1. If we graph our results, it should follow this sort of shape. The rate of reaction increases as temperature increases up until a certain point, which is the optimum temperature. Then the rate of reaction decreases until a stop. This is because at lower temperature, the particles have less kinetic energy, so fewer enzyme substrate complexes form. This is because the enzyme and substrate just don't collide as often. As temperature increases, so does the number of enzyme substrate complexes formed, up until the optimum temperature. Above the optimum temperature, the temperature is too high and the enzymes start to denature. The hydrogen bonds in the active site are broken, so the enzyme substrate complexes can no longer be formed. Anyway, thank you for watching. Please like and subscribe to follow the rest of our teaching videos. Follow us on socials to get some quick fire content so you don't have to feel guilty about your doom scroll. And finally, there's a link in the description to all the academic help we offer here at the Science Box. Thank you.